Okay, good afternoon, everyone. We are discussing about eukaryotic gene regulation. In that, we are studying about some of the epigenetic mechanisms. And one of the ways how epigenetics is different from the other type of gene regulation is other type of gene regulation. When we say it's about transient, in the sense that say for example there is an E. coli cell and you have lactose as a carbon source, then it should take it. For that, it will have an expression of lack of it, right? And if you remove the lactose from the outside, or the lactose, that the supply of lactose is exhausted, and in that case, the lac promoter, or there will be transcriptional repression. So many a times that we discuss the regulatory mechanisms, even eukaryotes that we have discussed about GAL4 and so on. They are within a life cycle from within one cell's lifetime. Within that lifetime, there will be responses to the external stimuli, right? Supply or starvation and those kinds of things. What if there is a way of regulation where some change has happened in the cell, and that regulation is the same response, or kind of some change is inherited also by the daughter cells and their daughter cells. It is something like you are having a response to a stimulus. This cell had a response to a stimulus. It could be something like a morphogen. Say in cell biology, you must have learned about morphogen. It is simply a biochemical or a compound within a growing embryo that will result in that will or induce some kind of morphological changes within the cell, right? One cell, because of some morphogen, it could change into or differentiate into a different type of cell, right? And those are those kinds of mechanisms where the response of a cell is inherited by the daughter cells that are coming out of it, or emerging, or belong to that lineage, are called as epigenetic mechanisms, right? This way we are trying to differentiate how epigenetic mechanisms are different from the other types of genetic mechanisms, right? So that is also important because especially in case of eukaryotes, and especially in case of multicellular, highly complex organisms like mammals or any other, say, humans. Say a zygote has become blastocyst. In blastocyst, you have ICM cells. ICM will give rise to, say, primordial germ layers. And there is a sequence of events, right? Ectoderm has come. It's like, imagine what would have happened. I will try to describe the same thing in a different way. And that way you will be able to also appreciate the importance of epigenetic mechanisms. Say, for example, zygote. And zygote has grown into a lot of cells, morula, and then becomes the blastula stage, where you have trophoectoderm cells and ICM cells, inner cell mass, which consists of several cells. We call them pluripotent stem cells, or they are the sources of embryonic stem cells. Anyway, the point is, assume that one cell of it has contributed, or the ICM cells. Okay, say this is inner cell mass, several groups of a group of cells. They differentiated into, I will write it as ectoderm and mesoderm. Some of them became mesoderm, and some of them became endoderm. Say, for example, one cell that has become endoderm, right, primordial germ layer, if it can happen two things. One is it can become back to that of properties belonging to the inner cells, or it might change it to become a mesodermal cells or to become ectodermal cells. And if this is happening, you cannot have organized differentiation of a cell mass to give rise to a complex or an organized body like this, right? So the pattern, differentiation pattern is strictly regulated within organisms. I hope you understood that. So those that have become ectodermal cells will not give rise to mesodermal cell types. That is not going to happen. They have a path and they will take that path and give rise to these kinds of differentiated cells. Right? That is how, because of that epigenetic mechanisms, the cells are going to follow a certain pattern, and because of that pattern, they will give rise to different functional cells, and then you will have an organized body as an organism. So that is one another way you can think of epigenetic mechanisms, right? We have also, this is a quick review of what we have already seen. There are several mechanisms, out of which DNA methylation, histone acetylation, chromatin organization, and nucleic architecture are some of the ways. DNA methylation and histone acetylation are the best understood at this stage. We discussed about cytosine and methylcytosine. This is the methylation that happens at the cytosine and in CpG islands. And we also discussed why methylcytosines are hotspots for mutations, because deamination of methylcytosine will give rise to thymine. And thymine is a normal nitrogenous base within DNA. And then there is more probability that this thymine will be retained, and that establishes a mutation. We also discussed about how DNA methylation can influence the gene expression, not only just influencing the gene expression, but it can also induce, upon methylation, it can induce the binding of several methylated DNA binding proteins, which could turn off much more efficiently. Yes.
Sorry to disturb, sir. Like some music is keep on coming from your side, and it's loud.
Yes, sir.
Thank you, sir.
Chill. Is it off now, or it's still? Is it off?
Yes, sir. Yes, sir.
Okay.
Thank you, sir.
Good. Okay. So, yes, I was here. So, it is just a DNA methylation that happened at the level of DNA, but it can eventually bring about complete shutdown or silencing of this whole gene. And in that, you have two different histone modifications. So DNA methylation is resulting also in histone modification, and that is by recruitment of histone deacetylases or chromatin remodeling complexes and so on. So this whole genome will be shut down, and the daughter cells that arise from this cell, the signature will be maintained. And that means signature in the sense here is the regulatory signature or epigenetic signature, that this part of the region is methylated or silenced, and the daughter cells are likely to retain that signature, that this part of the genome should be shut down. And how can that be achieved is one of the ways is to shut down. So one of the patterns is one of the ways how this signature is maintained is by, I hope you remember hemimethylation like we have seen in prokaryotic replication initiation, regulation of replication initiation. So how is this pattern? Here is a DNA, methylated DNA. The C cytosine is methylated on the 5 carbon, 5th carbon. And when replication occurs, you have one old strand through semi-conservative replication and one new strand. And the new strands do not have any methylation. And because of this methylation that is present, there will be recognition by specialized methylases that are called as maintenance methylases, and they will bind to, or they recognize this cytosine, methylated cytosine, and they methylate the cytosine that is present nearby. So I hope you understood this. GC. On the others, on the complementary strand, it would be GC. If you have a methylation and this is recognized and a DNA methylase, maintenance methylase binds, it will automatically methylate this cytosine here. And throughout the genome, you might have several places GC, right? And so as is shown here, there is a CG here and there is a CG here. The same is the case. GC or CG depends on situation and context. So in one case, there is no methylation. In the other case, there is methylation here and here, and there are no methylations here. So how come maintenance methylases are only recognizing and maintaining, methylating only the cytosine like this? It depends on this. It depends on the methylation pattern on the parental strands. In here, the parental strand cytosine is methylated, so you methylate it here. The methylase will methylate this cytosine, and this CG will remain as such. They will not get methylated. So this way, the daughter cells that are coming. So you have, this is the parent cell, and these are the two DNA or genomes that are coming, which will give rise to two cells, and they both will have at that location the same methylation pattern. If you remember how the nucleosomes are disorganized and then recruited during replication, we have seen how some of the histones are reused and distributed evenly to the daughter cells, daughter DNA molecules. Then those histone modifications are also retained, and they are retained in the same locus, right? So the same way, you can also have the maintenance of histone modifications as well. So the point of it is, one cell has a modification because of the stimulus, and the daughter cells are still retaining that kind of things. Okay? Yeah, and this is also important. There is something called as transgenerational regulation. Okay, if you are interested, you can try that, and that goes even between, say, that is also a part of genomic imprinting and also in humans. I will use this case. So, say for example, you are this is an individual, and the individual is married to someone, and they have a kid, and this kid is married. This one eventually is married to somebody else, and they have kids. So, if this individual here, individual A, had some environmental, psychological, or some kind of stress or abundance, probably it can also be abundance, that will change or could change the epigenome of this individual's germ plasm or gametes, and that could be inherited by the daughters or the children, and that could even be inherited by the grandchild. Right? It's something like typically what people say. There is a nice saying like, "You are what you eat," right? And that's not necessarily or the only truth. The truth is also like, you are what your grandmother ate or grandfather ate. These kinds of um, observations have come from the worldwide, or some famines that happened in Netherlands and some countries. What they noticed is people that have undergone severe famine in one generation, the effects can be seen even in the second or third generation from them. So this is an interesting area of research. I will uh, I will share you that Coursera course. You might want to try to do it during your vacation if you have free time. Okay, we also learned about genomic imprinting. Genomic imprinting is allele specific gene expression. When you say allele specific, you're talking about paternal allele or maternal allele. That means if we have a gene A and gene A from, we are diploid 2M, so we have two copies of the gene, one inherited from father, one inherited from the mother. And assume there are also about gene B and so on. In some cases, only the paternal allele is allowed to express, whereas maternal allele is not allowed to express. In some other cases, you may have paternal allele to be repressed and maternal allele to be expressed. So that's one generation that is sex specific, right? I mean, in the sense, sex specific in the sense whether it is maternal or paternal. Just remember it as that some genes are expressed only from the paternal alleles and some genes are expressed only in the maternal alleles. What happens to the next generation? So this person has inherited, and assume the person that is having this one, these genes now, is a female. Then what should happen? And that female um, will have children, and they should have some imprinting pattern. So here, this person has A, B, two sets, right? A and B, like paternal, paternal, maternal. Right now, this individual has to make eggs. During oogenesis, what happens? I will take it as eggs, and usually from during oogenesis, you will only have the production of one egg cell, haploid egg cell, and the three are lost. Okay, polar bodies and so on. If we assume that, say, I'm writing the four ends instead of just removing it as polar bodies. In this case, you will have AB. Each of them will have AB, right? And which one should be methylated and which one should not be methylated? Say this individual is getting two sets, right? Paternal and maternal. And in that case, whether the same thing is carried forward or not. So what happens is what we will see. Assume this is an embryo. During gametes or primordial germ cells form very early in the embryo. That is, when we are even developing, then itself the primordial germ cells form, and they remain. They do not divide until puberty. In eggs, if in females, for example, even meiosis actually already starts, but it's arrested in meiosis 2 and will remain like that until the woman matures or puberty and until the egg is released or fertilized. Okay, we are not going to all those details. These are, as I said before, these are details for you to remember that there is a lot more in molecular biology and gene regulation, especially in eukaryotes. So you have to be open to many different types of things. Okay, in imprinting, we discussed about erasure. Whatever methylations are there, which is causing the repression, they're all erased. And during gametogenesis, new imprints are made based on the sex of this individual. If this is a female, then it will have the methylation pattern. All the gametes that generate from this female will have the same type of methylation patterns or imprinting patterns. And that will, upon fertilization, will become biparental, and then they will have it. They would have inherited the imprinting pattern. For simplicity, what you should remember, right? Say you can take, we are all individuals, we have, our body has somatoplasm and germplasm. All the somatoplasm actually belongs to our parents. Genes and genome are imprinting patterns of our parents. They're not ours exactly. What is our signature in our body is actually the germplasm. In terms of, at least in terms of genomic imprinting, that is true. The events, many things that happen, the imprints that our parents have made, we are bearing them in our somatoplasm. We erase them even when during embryo stage, and we make a new imprint based on our sex. And that we will give to the next generation. And when the next generation form, their somatoplasm is that belongs to us, and they have their own germplasm again. And this cycle keeps on going. Okay. If it is complex, it's all right. You have a lot more time in general, to study, and I'm not going to bother you with detailed questions on this. But you should know what is genomic imprinting. We also learned about H19 and IGF2. This is a classic example of genomic imprinting and also epigenetic regulation. Say this is the maternal allele and this is the paternal allele. Maternal allele, H19 is expressed and IGF2 is repressed. And in paternal allele, H19 is repressed and IGF2 is expressed. How is this accomplished? It depends on DNA methylation. Every time only this methylation in this region of the paternal chromosome you have methylation. This is imprint control region where if methylation is there, has occurred, this is an insulator. So CTCF cannot bind in this case. So the enhancer will activate the expression of IGF2. But in case of maternal allele, this region is not methylated by DNA, or DNA is not methylated, sorry. Therefore, the CTCF binds here and it will prevent the interaction of this enhancer activator with IGF2. Therefore, the enhancer activator is going to activate the expression of H19. So, although in the same cell you have 2 copies of H19, you have 2 copies of IGF2, but only one X or only one H19 is expressed, or one IGF2 is expressed. Okay, so that is what is about it. So in today's class, I would also want to briefly describe about regulatory RNAs, some of them, and we'll finish this lesson here. So there are several RNA molecules, as I said. If you are into core biology, you can think of epigenetics, and then there is the other one is RNA biology. If you are, everybody that are going into core biology will have to deal with molecular biology. There is no escape from it, and your success will also depend on the strength or the foundations of biochemistry, cell biology, and molecular biology. That's the truth of it. So RNA biology is another important area of research because it's involved in many things and the diversity is huge. So there are several RNAs that can regulate, that can function in gene regulation. Here we are discussing only in that. Say for example, small RNAs like this, you can have, if there is a case like A, where the ribosome binding site, there is a mRNA mRNA has this open reading frame or coding sequence, and there is a leader region or 5' untranslated region. And in the same, you also have ribosome binding site. Sometimes there might be that this leader region or the 5' untranslated region may have complementarity with the mRNA, like is shown in here. And if that happens, the ribosome cannot interact with the ribosome binding site, and therefore the translation of this gene may not happen. And there could be some RNA molecules, short ones, which may have complementarity with this region. And if this region, 5' untranslated region, is base pairing with the siRNA, then the ribosome binding site is now free of it. Therefore, translation can start. Okay. You can see a contrasting example where ribosome binding site is there, and ribosome would have bound here, and translation would have resulted in protein and gene expression. But if the siRNA has complementarity to the ribosome binding site, it will prevent the translation from happening. And then that is going to result in inhibition of expression. So that is some of the ways. So if this is gene ABC, say, it will be expressed. So I will talk of it in the significance. Say this is DNA. Here is the promoter, and you have the mRNA produced, splicing, everything happened, and then this is the case. And say the ribosome binding site is here, and it is bound like this. Transcription has happened. Is translation happening? No, translation is not happening. The translation of this gene ABC would have occurred if only there is also translation or the production of the siRNAs. Then siRNA can interact and stimulate the production of the or translation, and then the production of the protein, which will result in a phenotype. Okay. There are other ribosomes, other RNA molecules, the regulatory ones, which are called as riboswitches. Riboswitches typically have several regions, especially the aptamer region and then expression platform. It's almost similar to what we have discussed previously. A little bit, a small difference is over there. The expression of this gene, say XYZ, will depend on the three-dimensional structure of this region. Right? There is a long or considerably long 5' untranslated region, which can fold into, based on its base pairing, which can fold into several hairpin loops and structures like this. And that is called as an aptamer. Aptamer is something that can bind to some other ligand. A ligand here is a small molecule. RNA can always bind to its complementary RNA molecule or DNA molecule through base pairing. This interaction is to a small molecule, and it's not about base pairing anymore. It is RNA molecule because it can fold upon itself, and that foldings may result in micro environments where they might be able to interact with a protein or with a small molecules, biochemicals, and so on, like amino acids, for example. Those are called as aptamers. So whether the expression, this is expression platform where the ribosome binding site is there. If the ribosome binding site and there is enough space for the translation to happen, initiation of translation can happen or not, will be dependent on the availability of the ribosome binding site or the probability of ribosome binding to the ribosome binding site. Okay, so what happens is if the molecule is bound, the structure is different. If the molecule is not bound, the the structure is different, right? They can change the base pairing because based on whether the ligand is bound or not, and based on the structure or conformation, whether the ribosome binding site or initiation of translation can occur or not. That is the essence of riboswitch. We call it as riboswitch because it's about aptamer, which is made up of RNA molecule, and whether it is turned on or off is dependent on the ligand. Okay. In different examples, you will find sometimes because of ligand binding, the translation might be inhibited. In sometimes, the ligand binding may activate the translation. It depends on the example that we are discussing. Say here, we are talking about one example. I would like to discuss riboswitch, which is activating, which is doing two things about regulating transcriptional termination or translational initiation. In example A, we are talking about this is the aptamer region, and this is the expression platform. So in this case, because S-adenosyl, it is for S-adenosyl methionine.
Please try again a bit later.
Sorry. So the aptamer is specific for SAM, or it's S-adenosyl methionine. And if it is not bound, you have several regions, 1, 2, 3, 4, similar to what we have discussed in attenuation into tryptophan regulation. Actually, that also is an example that would come into the regulatory RNAs, but we discussed it at a different location. Yes. So if the ligand is not present, 2 and 3 would form a hairpin loop, and transcription is allowed to proceed. But if S-adenosyl methionine binds to the aptamer, the binding pattern changes, or the hairpin pattern changes. The 1 here, which was free, and 2 and 3 are here. 1 and 2 would form base pair like this, and 3 and 4 will base pair like this. And formation of this hairpin loop 3 and 4, it's a kind of transcriptional terminator which will kind of induce transcriptional termination. And that is a riboswitch based on whether the ligand is bound or not, is able to regulate whether transcription should proceed or be terminated. The same example in the other one is B, and because of the same similar situation. What say, for example, the mRNA is already made, and in that case, what should happen? Should translation still be produced? Should still be proceeded or not? It's a waste. Every time, it's always important for the cell to not waste any resources, right? So it will also be important for if SAM is present, the particular mRNA should not be translated because it's unnecessary wastage of proteins and resources. So if SAM is not present and it is not bound, then 2 and 3 will form a hairpin loop, which will allow the ribosome binding site free and translation would occur and the protein is produced. Whereas if SAM is present and it binds to the aptamer, then you will have 1 and 2 and 3 and 4 base pair. 3 and then the 4th ribosome binding site is within the 4th region. And if the 3 and 4 form a hairpin like this, then the ribosome cannot initiate translation, so the translation is not, or the translation initiation is inhibited. So you can see that small molecule just and the organization of aptamer can decide or can influence the consequences of whether transcription proceeds or not, whether translation initiates or not. So there are several other examples, like for B12 vitamins, you can see there are several other compounds like this. Lysine, S-adenosyl methionine that we have discussed. And sorry, I forgot to mention this one, right? So this is how the banding, the base pairing patterns actually change. 1 and 2 and 3 and 4. We don't have to remember. It's the same thing. It's a close shot with the nucleotide sequence. So even for adenine, we have there is a ribose switch and so on. I mean, with more knowledge, we will probably know more details about how many more ribose switches are there. At this juncture, I would want to tell you this. All of life has started as some simple nucleic acid, probably RNA. That must have given rise to DNA. Or at that time, or before the evolution of cells, or after the evolution of cells, it could have happened. That from here we have generated, we got cells with, say, DNA and so on. We call it as universal common, last universal common ancestor. That means it is thought that all the life that we see at this stage in our day on Earth must have come from Luca, last universal common ancestor. From here, you will have prokaryotes and eukaryotes. Then, what I mean to say is what was in DNA, and then we have production of proteins, right? In this stage or so, we have DNA giving rise to proteins through transcription and translation. RNA. And do you have several types of RNA forming, several types of proteins forming, several structures that can form, several enzymatic reactions that can be carried out. And evolution is a continuous process, and more complex and sophisticated regulatory mechanisms or novel structures keep coming up, and that will keep on happening. If we go for in next billion years or million years, we might have even more things, amazing things that might have formed based on the context. So see it have a fluid and systems type of thinking when you're studying about molecular biology as information or in perspective of evolution. That's very important. So now we will quickly go to some short RNAs, and there are 3 different molecules which will not go into too much detail, but just know that there are small interfering RNAs, there are microRNAs, and there are pi RNAs. At least we'll have some description about small interfering RNAs and microRNAs. In the textbook, we also actually had about CRISPR. You heard about it. I remember some people presenting some poster about it and so on. That is a very interesting thing, interesting phenomenon to understand just as a biology, not necessarily as a technology and so on. That comes later. How did these systems evolve? If you start thinking like this, it's really fantastic. We do not discuss this in CRISPR in this course, but in our recombinant DNA technology in medicine, that's in the 7th semester. I have an elective there, I discussed that. But right now, we don't have it. Otherwise, it will become too much. Already having ribose switches and all these things itself is too much. So I will want to discuss something like this, the siRNAs, how do they form and what can they do, right? So RNA-induced silencing, RNAi technology says, they say, RNA interference technology. That is regulation by RNA, interfering RNA, and so on. We just see it as a process, and then we will see.
Please try again a bit later.
And then we'll see what we can do. So some RNA molecules are produced as long double-stranded RNA molecules like this, and there is an RNase called as dicer, which is the name for dicing or say cutting, and it cuts the DNA, this RNA, double-stranded RNA, into some molecules like this, right, with small overhangs, and they are about 20 to 20 base pairs approximately. siRNA is for small interfering RNA. They are then bound by a complex called as RISC, that is RNA-induced silencing complex. And the complex, so one of this will be discarded, and the small the smaller RNA, siRNA, along with this, will then bind to the target RNA. What is this target? Anything that is complementary to the siRNA. Okay, and if it binds here, and then risk, there are several things that could happen. The first of all is this is RNA dependent RNA polymerase. It can elongate the siRNA depending on. So if this is 5' 3', this would be 5, and here 3'. So more bases could be added, and that is how you can, by RNA dependent RNA polymerase, which will elongate it, and the dicer can act upon it again to generate more number of siRNAs. These siRNAs then can go and influence. What is that they influence? They have three different types of responses. One of it is to degrade the mRNA or the digest the mRNA. The other one is to interfere with translation of those mRNA. We will see in the next slide. And the third one is they can influence the chromatin modifying enzymes to come to that area and then change the chromatin structure itself. Okay. So what we have to remember here is these are long double-stranded RNA which are diced and made into smaller siRNA molecules which are bound by RISC complex, and they can be amplified by the RNA-dependent RNA polymerase, which will synthesize further, and more dicer comes again and will break it into smaller pieces. Now we have, for one particular mRNA, say mRNA of PQR. Sorry. Sorry. So there is an mRNA here, or PQR, right? And we initially had one siRNA, and along with this, it has come here. Excuse me. Okay. So the rest of the region is otherwise not targeted by siRNA. But how can one generate more targets for that mRNA? So if risk comes, this RNA-dependent RNA polymerase comes and produces more of this transcript, right? Now you have a double-stranded RNA. But yeah, and dicer might break this, and each of these RNA molecules can then be again bound by RISC complex and be used on other PQR mRNAs. Now you have this. The first one is only for targeting this part. Now we have this part, and this part of the RNA that are siRNA present to target those that particular PQR. So there is efficient way of regulation. It can result in degradation, it can result in translational inhibition, and so on. So I'll take this one slide and I will stop. So double-stranded RNA, that is the origin of siRNAs, and for siRNAs, miRNAs or microRNAs are different in the sense that they are produced as single-stranded RNA, but they form hairpin loops like this. So dicer the RNAs that breaks, it's a type 3 RNase 3 type like RNAs, which will polish it and break it into smaller pieces. siRNAs and mRNAs are processed in a similar fashion. They're produced differently, whereas they're processed in a similar fashion. And that will result in small fragments um, of, of double-stranded RNA molecules. One of it, the one that is going to bind to the target, is called as the guide RNA. Why do you call it the guide RNA? Because risk is there. Risk complex can only go and bind, can bind to, say, cannot bind to mRNA by itself. And where exactly to bind, it is all dependent on the sequence of the guide RNA. This guide RNA, based on complementarity, will allow the binding of the to the target at the where the particular sequence is there, complementary sequence. Therefore, we call it as guide RNA. I think passenger RNA doesn't have a much of a value at this stage, or knowledge is we do not know. And this get discarded. Sorry, the top one is the passenger that gets discarded, and the risk along with the guide RNA can have several uh, consequences. One of it is amplification. What we have seen here is kind of amplification. That means by involving RNA-dependent RNA polymerase, you can produce more siRNAs, and then you can target you can efficiently induce some effects on the target. Degradation, by binding of this, you can also have the slicing of the RNA, the mRNA. Therefore, you will not have any more of the gene expressed. Translational inhibition can happen if the binding of it has influenced or masked the ribosome binding site from initiation to occur. That is one of the other consequences. And the other one I told you, where chromatin can be changed. The risk, along with the risk complex, they can go here to the place where their gene is being produced, the complementary genes or the targets of these genes. They can bind there and recruit modifying enzymes, chromatin modifying enzymes, such that they will eventually become silenced, and therefore there will not be any more production of or transcription from that particular target gene. So these are some of the things that will. Okay, the others are all the same thing, but I will stop with this slide because this is what is going to explain how the gene regulation, how chromatin could be silenced. Here is a DNA, it's a repeat that is producing a double-stranded RNA molecule, which is acted upon by Dyson, and you have the production of siRNAs. They could be amplified by ribosome-dependent, RNA-dependent RNA polymerase activity. And that the siRNA could be bound to a complex like this, which is almost similar to that of RISC complex, except you have different other things called as you have RNA, RITs, and so on. This is the region that is being transcribed, right? So RNA is being produced, and if there is complementarity to the siRNA, again remember the siRNA is produced from that region only, right? So there is, you can expect that there will be complementarity, and if the complementarity is there, they will bind here. Once they bind here, they will recruit several other enzymes, chromatin modifying enzymes, which will result in the methylation or deacetylation of the nucleosome histone scheme, and also induce methylation. You can think of these flags are about acetylation. Okay, here it is. So if histones are acetylated, which is usually associated with gene expression, if they are deacetylated, they are not expressed. And in some cases, if there is methylation in certain residues, then there is also repression. So here you can see that siRNAs are produced, they are causing they are recruiting several proteins that are required, several enzymes that could change the histone tail modification signature, and thereby what was actually being expressed is now repressed, and that will continue to be taught to cells as well. Okay, so there will be a lot of names and gene names, protein names, and so on. I don't need you to mug up these things because it's every time you read, you have to always look into this. I would rather want you to understand the evolutionary perspectives of that and the physiological relevance of these concepts and the logic of how things happen. Okay? So make more brain space for principles rather than for every detail of a mechanism. Right? I mean, I understand that if it is an exam, you will have to, of course, mug up a little bit of these things, but principles are more important, I think. Okay? Because principles recur in one cell to different cell. The same principle may happen, but the names of the enzymes and genes may change. That's why I would want you to focus more on the principles of things. Okay, let's stop the recording.